given your kind of two-country background, I think one of the most German things you could have done is go study in the Netherlands, at least for someone who, I mean, I guess, yeah, we established before we started recording that we grew up somewhat close to each other. And the people, I mean, Aachen is right on the border to the Netherlands. So I don't know, 10% of the people I knew studied in the Netherlands or something like that. But why did you study in Leiden or in the Netherlands in general? And yeah, what was when you were in school, what made you go there?
So I'm going to be brutally honest, and you have to understand that I was 17 when I finished high school. And so a huge reason why I decided to go to the Netherlands is because I thought that going there to study there and live there would be just like my parents would take me and my sister to the Netherlands on like short trips over the weekends. And obviously that was always a blast. So I thought that everyday life in the Netherlands was like that, and that my life was just going to be that, running around the beach, catching jellyfish with a little catcher. So that's why I wanted to go to the Netherlands.
And Leiden is at the beach, right?
It's very close. Yeah. So yeah. And it is stunning. So it really, in some sense, was very beautiful to live there. But I mean, Leiden in particular, when I always wanted to study biology, but I wanted to do working with animals biology. I wanted to go into the jungle and go find some new animals and new plants.
Right.
And Leiden is really strong in their ecology departments. And they actually still have a lot of scientists who they work very closely together with the Natural History Museum. So they have a lot of scientists who literally go to Borneo mostly every year and find some new insects and new plants. So I wanted to do that. Yeah, it is, yeah. So that's why I decided to go to the Netherlands, and that's how I picked Leiden.
Okay, but I don't know, I guess you could have done the same in Cologne or something, right? Or did you also, was going to a different country also part of the appeal of leaving home? And I mean, that's why I went also, I mean, for me, there were multiple reasons why I went to England, but one of them was also just like leaving. Not that I had like a bad time at home, right? It's not like that kind of leaving, but...
No, I understand. Yeah, same. I wanted to go somewhere new, but I was also 17, so being able to go to a new country, but still being
not
2
too
hours.
far away. Yeah, exactly. Being able to take a direct train home, for like 30 euros, or whatever.
Exactly. Yeah.
Okay.
And I completely ignored the fact that Leiden, at least at the time, taught all of their STEM undergrads exclusively in Dutch. And I didn't speak any Dutch.
But that's not too difficult, right? I thought you just... I know some people who took like an intense Dutch course for a few weeks and then they got worked.
Yeah. I mean, that's what I thought. I definitely struggled the first year, but eventually I got the hang of it. Yeah. And it worked out.
Okay. But so what was the degree? Was it like so my sister briefly studied biology and they had like all these, you had like these leaf collections that you then dry, and did you have to, was it all that kind of stuff, or learning all the insects and whatever? Yeah,
so in the first year, the biology undergrad branches into what they call macro and microbiology. And so microbiology is more bacteria and genetics, and macro is basically plants and animals at the plant and animal scale. And the first year is half a year of each. And then you have to decide between the two. And so I went in thinking I'll definitely do plants and animals. And then the half year of plants and animals hit. And it was we had an exam where they would literally give us a bunch of dead animals, mostly insects and plants. And we would just have to identify them by their Latin names.
Nice.
And that is when I decided that maybe I would go in the microbiology direction after all.
Okay. And in hindsight, do you think that was a decent decision on your part, or was that too reactionary to the specific environment you were in there? Because I guess that does suck, yeah. That doesn't sound like a fun time.
I think it was. I mean, I don't know what my life would look like right now if I had not done that, if I had gone the other way. But I'm pretty happy where I am now. So that worked out. And I do think, I mean, there's absolutely more opportunities in the microbiology, especially genetic space. There's just so much I can do. So I think in that sense, I like that I'm not limited in my work in the way that I think I would be if I had gone the other route.
Yeah. Did you want to do research from the beginning? Or
Yes.
Okay.
Yeah.
So it wasn't just like animals or whatever. It was you want to do research and.
Yeah, I always wanted to do research. I was really fascinated by this idea that there would be this moment in the lab when I would be the only person knowing something that nobody else knows yet. But I just like figured it out, so I know it. So I've always really liked that idea.
So how often has that happened, proportionate to the rest of your working hours?
I don't want to discourage our early career listeners too much. It's been really rewarding. I don't think that moment is as specific. I don't think in the moment you even realize that just happened. So yeah,
that's a good point. It's more like afterwards you're like, wait a minute, these things I'm piecing together here actually amount to something.
Exactly.
Or you already amounted to something and I didn't realize it.
Mm-hmm. Exactly.
Yeah. I think there's a lot more. I mean, any job, right? There's more drudgery than the highlight moments.
Yeah, there's very few eureka moments in science.
More pipetting than eureka. Yeah.
Yep.
Although, funnily enough, I talked to Jessica Polka. She did a PhD in, I don't know, something molecular biology. I mean, we talked about preprints and open science and that kind of stuff because she works in that field now. But she actually said she misses pipetting because there's a kind of nice, like, manual labor to it that she just doesn't have anymore.
I prefer gardening, so I do not miss the pipetting, I have to admit. Not now. I mean, maybe I will in a couple of years.
Yeah, maybe we'll get there. Okay, so you wanted to do research from the beginning. Did you, I'm assuming you did some sort of research project for your bachelor's and then later for your master's, or?
Yeah, so after I decided I was going to do microbiology, I had to figure out again what I wanted to do. And mostly because of a single lecture by a professor called Egbert Lacke. He's also a neurologist at the Leiden University Hospital. And he gave a lecture on vision and how visual input is decoded in the brain. And I was so fascinated by that. I was like, I want to do neuroscience. You'll see throughout my career, I've had a lot of these moments where I make really important decisions based on what you would think is not a lot of data, but it always worked out. So in the end, it always worked out. But so basically, I decided I wanted to do neuroscience. I was still in a genetics program, but that was fine because there's a lot of need for genetics technologies in neuroscience, naming optogenetics, just for an example. So the problem was that Leiden University basically doesn't do any neuroscience. They do neurology at the hospital, but that's it. They do psychology, but they don't do molecular neuroscience or neurobiology like I wanted to do. But it turns out that also worked out because that forced me to do all of my internships outside of Leiden. So I did my first internship at the Max Planck Institute for Neuroscience in Florida, which was awesome because it's right on the beach.
To have a Max Planck outside of Europe? Okay.
Yep. And it's right on the beach. It is absolutely stunning. I was there for 2 months and I think I did 60 scuba dives, which is basically a dive a day.
One a day, yeah.
So I had a blast, and that was also my first really serious. I mean, I was a first year, but I was in the US. They're not that cautious with rules around who is allowed to do what. So I was allowed, even in the just 2 months that I was there as a freshman, I was allowed to work with mice. I was allowed to learn mouse surgeries. I was allowed to do all of their cloning. And so, I mean, I was just a lab tech for the postdoc, but I learned so many very valuable laboratory techniques that in the Netherlands, I would not have been allowed to work with mice because I didn't have my bachelor's yet. So that was amazing. I learned a ton, and that was my first really serious wet lab exposure.
I mean, so that's also a lot of trust into someone who then, I guess, were you 18 at the time then already? Or were you still 17?
I think I was 19 because I had
I was a bit bigger.
I had to do 1 year of chemistry and physics because there's a mismatch between the the Dutch and German STEM system, where in the Netherlands, you have to have studied biology, chemistry, and physics to study any one of them. But in Germany, you're not allowed to do all three in your Abitur. You have to pick two. So I didn't do chemistry, so I had to redo chemistry, and then I thought I might as well do physics as well.
Okay. But still, I mean, that's a lot of trust to put into someone who's also going to leave in like 3 weeks.
Absolutely, yeah. Yeah, I'm super. I mean, now that I have a bit more of an understanding of how a lab is run, the amount of resources that I was entrusted with, I am really grateful for that opportunity. So I don't know if the professor or the postdoc are listening. The professor was Hiroki Taniguchi. If you're listening, thank you. Yeah, it really made a difference.
Okay, yeah, I mean, it's cool. I mean, I guess I also don't know like how difficult or complex these things are. I mean, in psychology, when you like a research assistant, you basically, your main job is just, I mean, it's like a job any 12-year-old could do most of the time, where it's just, you just take someone, put them in the room, say, here's a computer, the instructions are on the screen. And then when they're finished, you pay them. And if you're vaguely pleasant, then you're doing a good job, basically, most of the time.
No, yeah. I was doing in utero electroporation, where you cut open a pregnant mouse, inject DNA into the brain of the embryos while they're still in the uterus. And then you have to put everything back. And you have to do, this is a very delicate surgery because you have to do all of that. If you stress out the mouse too much, they will spontaneously abort. And so, yeah, I felt so cool. I was like...
But I mean, do you think that's a good system? It's obviously amazing to, if you want to be a scientist, it's amazing to learn all these things. But it seems to me it's weird that, especially because you were only there for a short time, it's a lot of training resources, a lot of animals and that kind of stuff for someone who is going to leave soon anyway. I don't know. It's very surprising to me. Yeah.
Yeah. I mean, I will say, they were not lax about how they handled the animals. So I did receive a lot of very intense one-on-one training in a short period of time. I think it helps that the Max Planck Institute is very small. So I had the animal technician to myself, and I had the postdoc to myself, and I had the microscopy facility manager to myself. And so I could talk to really the experts for every type of procedure that I wanted to do, and they had time to train me one-on-one. So, but yeah, like I said, I am really grateful. The benefit to them was not that big compared to the benefit to me, which was huge. Yeah.
Yeah. Okay. So you did some surgery, you did some scuba diving, then you go back? Or...
Then I got back. Yeah, I came back, finished my bachelor's. I did my bachelor's thesis at the only place in Leiden where they have anything close to neurobiology, which was the hospital. So I was analyzing but this was now in the Netherlands, so I wasn't allowed to do anything in the lab, but I was just analyzing the data, which was also great. I learned a lot. And this was an optogenetics project on trying to understand a very specific type of migraine, which can cause sudden death in children. So we were trying to figure out the link between the migraine and the sudden death syndrome.
Okay, that's pretty cool. It sounds like a pretty cool, many bachelor's projects are not that cool.
Yeah. No, so I loved it. I was like, okay, neuroscience is my thing at the time. And I really enjoyed going to different places to do my research. So I actually decided to stay in Leiden for my master's, even though, again, Leiden doesn't offer a neuroscience master's. I, again, did my master's in genetics. But the reason I stayed with Leiden is because they are extremely flexible. So I was able to basically not be there for 1.5 years of my 2-year master's in Leiden.
But why not just do your master's somewhere else immediately? Or was it because you wanted to try lots of different places? Or
I really wanted to do a lot of research. And so the flexibility of the program, I was able, usually the program is 1-year classes, 1-year research, but there's a way that you can shove all of your classes into half a year, which is what I did. And then I had 1.5 years where I could just do whatever research I wanted. And so that flexibility is what I wanted, is really to have all of this room to do research and do it wherever I want.
So you did some neuroscience?
So I did some neuroscience. And I did 2 projects. So I wrote 2 master's theses, both of them neuroscience projects. And one of them was at a company because I wanted to see a science environment within industry with brain organoids, also very cool. But the other one was at Caltech in a neuroscience lab studying how antidepressant drugs work, or trying to figure out, basically we were building sensors for the antidepressant drugs so we could follow them as they enter our bodies, as they enter our cells, and see if that localization tells us something about their mechanism of action.
So one of the obvious questions I had was, whenever someone jumps from one place to another, is, oh, well, how did that happen? So that's how the Caltech happened.
Yeah.
You liked it there and then just decided to apply to stay, basically?
Yeah.
Or
yeah, pretty much. So my internship at Caltech was 10 months, and I wanted to do a PhD, but I was planning to do my PhD in Europe. And as my 10-month internship was going to an end, this was like a month before applications are due in the US, my professor was like, you should apply. Why not apply? You could do your PhD in the US. You could do it here. And you have to understand that PhD applications in the US, they're not like PhD applications in Europe. I think in Europe, you apply to a professor directly, so you have to adhere to their requirements.
There's also programs. It really depends.
Yeah. I think the
Often,
institutes, Max Planck, they have more defined application cycles. But in the US, you have to take the GRE, which is a very painful exam, 4 hours. You're not allowed to go to the bathroom. You're not allowed to drink anything. You just sit there for 4 hours and have to take this exam. And then you have to write 2 or 3 essays about yourself and about the science you want to do, and then a very specific proposal. But I didn't really know all of that. So I was like, I'll apply. And then I realized that I had a month to do all of that. And I don't know how well I did. I guess good enough. I got offers from Caltech and also from Johns Hopkins.
Sounds like you did something right.
Yeah, I did something right. But the GRE was really painful. So I think now a lot of universities in the US do not require it anymore, which can be a pro and a con. For some people, it's a nice way to show their skills. For me, with a month of preparation, it was just painful.
But I guess then it's also over quickly, right? It's not like this protracted process that you prepare for and blah, blah, blah. It's just you get it done with and then... Yes. I mean, when it works, it's nice. Obviously, if it doesn't...
Yeah, exactly. Then, in this case, it worked out. And I got the offer from Caltech unexpectedly. They flew me out to interview, and that went well as well. So when I got the offer, I thought, this is one of the best institutions of the world. I can't really say no. So that's how I ended up at Caltech.
Okay, but you still had you ended up doing your PhD with Lior Pachter. Is that how you pronounce it?
That's right. Yeah.
So, but it wasn't clear to you that you were going to work with him or anything like that. It was just, you liked the place and so let's stay here and see what happens, that kind of thing? Yeah. Oh, okay.
Yeah, I think throughout my career, I've been doing a lot of hopping around, which I think is clear now. And so I just always had a lot of things that I'm interested in, and I can get excited about pretty much any project. So when I came to Caltech, I had the same mentality. I liked that in the US they have this rotation system where in the first year of your PhD, you do 2 months in 3 different labs and then you pick one. I like that because, I didn't have any one thing that I was really set on doing. I had a lot of different skills that could be applied to a lot of different research areas. And I found it really difficult to pick one. So I went into Caltech with the mentality, something will come along and I'll know.
Yeah, that's, I think let's maybe talk a little bit more about that, because I don't think you're unique in that particular thing. I think I also ended up doing lots of different projects, getting involved in research here and there and doing all sorts of stuff. And what I ended up doing my PhD in was nothing I did basically before. And so maybe how did you, how did you basically end up making the decision of, given that there presumably were quite a few options available to you at Caltech? Yeah, how did you make that decision? Because it's a decision basically everyone has to make at some point, and I think many also struggle with it.
Mm-hmm. So I do want to start. So when I first came to Caltech, it was split. There were a lot of people like me, like Lasse mentioned, but then there were also a lot of people who had really basically worked in the same lab their entire undergrad and career after that. So they were really experts in like very particular fields. And that was very intimidating. And they really, they knew what they wanted to do and they were already really good at it. And so I often felt like I wasn't good enough. And I was like, how am I going to compete if any lab that I consider has one of these people who literally trained for years to join their lab? But I think that in the end, the flexibility that I had and also the skill to very quickly learn a new topic, which I had acquired by just switching around, having to do it, that was super, super valuable. Because anytime there was an obstacle, I was able to adapt and work around it and find some interesting angle, even though it wasn't in the original frame of what I had pictured for the project. And that flexibility, I think, was harder to obtain for students who had always been in the same lab their entire career.
To just briefly do a slight mention some of the stuff we'll talk about later, is that, for example, the thing you then was it gget, what we'll talk about a little bit, is a software program in genetics and not in neuroscience per se, right? So it's two different fields that you didn't plan on working on.
I completely pivoted again during my PhD. And so I can talk about that because it's also how I ended up in the Pachter lab.
Yeah, perfect then.
Yeah, well, one of the goals I had for my PhD, and that's also why I chose a very technical school like Caltech, is I wanted to learn programming. I didn't know how to code. I think Leiden University does better now, but at the time they just didn't teach their biology students how to code, which is just not acceptable anymore. That data that we're generating today as modern biologists, you cannot plug them into Excel anymore and just make a plot in Excel. They're way too big. So I really wanted to learn how to code. And when I started at Caltech, I took every class that I could find that I thought I would understand, which turned out to be not a lot, because even classes that said doesn't require any coding, they always required coding. But I spent a lot of time on YouTube. I spent a lot of time on Code Academy. Learning to code is really painful in the beginning, but it is an exponential curve. So anybody who is struggling through that right now, it's very frustrating in the beginning, but then it becomes really fun. So I struggled through that, and I found that I really enjoyed coding. And then the pandemic hit. I started my PhD in 2019. So I really only had half a year, and then we were locked in at home. So the pandemic hit.
I had the exact same thing. Yeah, exactly.
Exactly. I just couldn't go into the lab. And so I think this is where a lot of students really struggle, understandably. It was a very difficult time to do your PhD. But I decided, well, I just want to code. And now I'm stuck at home anyway, so I'm just going to do that. And at the time, I was working with another student who was generating a single-cell RNA sequencing data set. So very complex, very high-dimensional RNA sequencing of thousands of cells. So you absolutely need coding skills to work with a data type like that. And I was the only person in that lab who knew how to code. So she came to me and...
Wait, but how... I thought everything was so technical and required coding. How were you the only person who knew how to do it?
It's very technical in terms of classes. So all of the Caltech undergrads know how to code, but the PhD students and postdocs are very international. And so a lot of them come from universities, especially in the biology labs, who are still a little bit behind in teaching.
It's funny that the undergraduates are, in some ways at least, more skilled.
Certainly in coding. I mean, right then they lacked obviously the
That's other stuff, but just for coding.
I mean, Caltech does a really great job in teaching them technical skills. They are very good programmers.
Yeah. So just a general question about, because I'm not from molecular biology, and I mean, I guess technically if you do neuroscience, neuroimaging and humans, I guess that's a kind of biology, but I don't know. Biologists probably don't like it when you say that, I could imagine. But I'm still a little bit surprised that it's that rare, because I feel like if you do any kind of neuroscience in humans, fMRI or EEG or anything like that, or even if you do most cognitive science, everyone has to code. I mean, I don't know whether I know anyone who didn't have to learn in his PhD, basically. I mean, I don't like it. It's not my favorite thing. But yeah, I'm just surprised that like in biology, it seems less common.
Yeah, I think it might also be the exact subfield that I was in. Maybe, I mean, I was trying to do wet lab neuroscience, which is very heavy in the lab. So you just don't have time to sit down and learn how to code, doing a lot of cloning, a lot of cell work. And then the main output is microscopy images. And so Fiji is still a very widely used program to just analyze those images. Sorry. Fiji.
Other than an island and a water brand. So
there was a very popular image analysis program among biologists that generate microscopy images. And that program was called ImageJ. And since it was so popular, they kept maintaining it. And then they put out a new version called Fiji, which stands for Fiji is just ImageJ. And so anyway, ImageJ is, you can imagine it as the most kind of minimal user interface. Think like 90s computer style. No. It's just a little toolbar, and you basically give it your microscopy image. And then you can actually do a lot of transformation. So it is absolutely a very well done, very valuable program. But obviously it's not able to handle high throughput analyses in the way that, especially very specific individual analyses, in the way that you can if you write your own code. So everybody in my lab was just throwing their microscopy images into Fiji, a lot of Excel, and that kind of worked out. Yeah. But yeah, you really can't do that with sequencing data, because now you're talking about millions of reads in thousands of cells across tens of thousands of genes. So obviously you can do that in Excel. So yeah, she approached me to do that. And I was like, sure, I can do a single cell sequencing analysis. It turned out to be a lot harder than I thought it would be. I think it is absolutely true that analyzing a single cell RNA sequencing data set properly is a PhD project in and of itself, regardless of the data set. And so I think that right now the system where usually the biologists themselves are expected to just somehow know how to analyze this like really complex, noisy, high-dimensional data it's a bit messed up, and we're really asking a lot from our biologists right now. And so I also think that biologists who do that are not getting enough credit. I think bioinformaticians can get very specific and they critique biologists when they make mistakes in their analysis. And I think that the fact that they're doing the analysis at all is really impressive. So
I guess it's similar in the stuff that I did, that you basically have to do so many different things that of course you're not going to be great at all of them.
Yeah. But yeah, so I had absolutely no idea what to do with this data. So I reached out to Lior, because he is the expert at Caltech in RNA sequencing, or even, I mean, he's a world-renowned expert in genomics. So he was the obvious person to reach out to. And I just sent him an email and I asked, look, I'm getting this data set. It's really cool. I really want to do this, but I have no idea what I'm doing. Can we just meet every couple of weeks and I present to you what I did and you tell me if it makes sense or not? And he said yes. And so that's how we started working together. And I decided that I loved it. I decided that I found out that my deep biological knowledge was actually extremely valuable for this field. I mean, Lior is a mathematician. His lab is mostly mathematicians, computer scientists, physicists, some chemistry. He actually has a wild mix. He also has some biological engineering there, but a lot of more technical degrees. And so then I came in with very little technical computer science knowledge when I started, but really in-depth understanding of genetics, of neuroscience. So in this case, it was a neuroscience data set. So I really understood which questions to ask. And maybe my code to answer them was a little sketchy, but it worked. And if you don't know the questions to ask, your code can be beautiful. It's still not really useful. And so I found that there was really a niche there for me that I just, really enjoyed.
It seemed like your role switched then from you didn't know that much biologically, but you knew how to code, to suddenly you're like the person who knew nothing about coding or formal analysis, but you have all this biology knowledge.
That's right. That's correct. So my PhD in the end has been completely computational. There's zero wet lab work that I have done myself in my thesis. And I'm really happy with that. I really enjoy the work. I really like being this bridge between biology and bioinformatics. I think there's a lot of need for that. Actually, I mean, that's how gget started.
Yeah, I was going to ask, so like how, I mean, maybe also I'm assuming that most of my audience, based on the people I've invited so far, don't know that much, or at least are roughly like me in that they don't know that much about most things, but biology in particular, maybe, or like molecular biology. So maybe what were you trying to do? What was the problem for which you started, I think, as first somewhat accidentally developing and then later a bit more systematically?
Yeah, so let me first describe what gget is, maybe. So basically, it's a software that allows you to very easily pull specific information from large public databases that contain genomic information. So we have a ton of genomic information out there. Think, for example, the entire human genome. You probably all know that it's sequenced, and you can all look at it. It's all public information. You can literally just go to a website called Ensemble, and you can go look at the human genome and look at all the genes in it.
Just read it.
Just read it. Yeah, you can literally just read it. I don't know if I would recommend it, but you can. But you can imagine that, right, we have amassed in the last 20 years a ginormous amount of genomic information. We have not just the human genome, but the genomes of many more species than that. And even just for the human genome, there's DNA, then there's RNA, which then encodes proteins, which have different functions, and all of that information, and also different versions of it, and also genomes created by different groups, all of that information is stored in public databases. And it's extremely important reference information when doing, for example, an RNA sequencing analysis. It's great that the information is public. However, it's not always that easy to actually then access it, because every one of these databases has a different website. You get immediately bombarded with a ton of information, a million different options. So it's actually not that easy to find exactly the information that you need.
So just briefly for context, like what kind of information would I be looking for? I mean, so genome, I'm imagining just like a bunch of CG, A, and Ts or whatever. And like, how do I look for something? What's the,
I think that the easiest example is you do your RNA sequencing analysis, which means that you have your RNA sequence, which is a bunch of AC, Ts, and Gs. You align it to the human genome. So then hopefully you know what your RNA was, what function it had. Mm-hmm. So getting the human genome, that is one thing that we need to do from those websites. And then also, once you know what your sequences are, once you have the gene names, that doesn't tell you much about its function. It's just a name. So to then go from that to a biological conclusion, you need to learn about the specific genes that you're looking at. biologists don't just randomly know what all 60,000 genes in the human genome do, and different combinations of them might have different functions. And so all of that meta information on the genes that you might be finding in your data, all of that is in these public databases. And that is the type of information that I think is most often accessed. So you already hinted at it. Yes, all of the information is there, but it is quite difficult to access. So all of these websites have different interfaces. You have to learn them. You get bombarded with different information, 99% of which you're not looking for at any given moment. And so basically what gget does is you can give it a gene name, and it will pull the information about that gene from all databases simultaneously. And it's very specific information. So I really leaned on my knowledge of the information that biologists needed here to make a selection of exactly which information I wanted to emphasize and pull out. And so that's how gget started. That was the first thing that gget did, is facilitate access to these websites so you can very easily pull certain parts of information from all of the websites simultaneously without having to access each website manually and try to learn and navigate and do all of that for hundreds of genes, right? That's how gget started. Now we have, I think, over 16 different modules, so sub-packages of gget that each do something else. And we have over 120,000 downloads all over the world, which is really cool to see. So it really hit a spot. I think a lot of biologists were struggling with trying to access this information quickly. And I think that, specifically for gget, this knowledge of what biologists need in their analysis, it's now also widely used in protein sequencing analysis. I wrote a module to run AlphaFold 2, which is a very famous protein modeling. I really had to think very carefully about how I wanted to design gget so that it wouldn't have the same problem these websites have, where there's just too much, and that then makes them too difficult to use.
So is one of your problems just websites? So it seems to me really that it really is like software. It really, first when I heard about it, I thought it was, how should I put it? I mean, it really sounds like a way of using software to automatically just get information that you want from the web. So is one of your problems that these websites just change their, the way they set up their structure or something, and then you have to reprogram everything, or is that?
Yeah, that happens all the time. So we have bi-weekly tests that run and check every single call that I make to any of the servers to make sure it remains stable. And I capture problems, I capture bugs and their new releases in my tests all the time. So yeah, it definitely, gget has grown out of that. So for example, for the AlphaFold 2 module, the company who made AlphaFold, DeepMind, they were so proud that they were like, oh, our code is all open source, anybody can use it. But then you go to the GitHub repository and it's, you can use it, but you need a modern NVIDIA GPU, and you need to be able to work in a Linux environment, and you need 2 terabytes of disk space. And I'm like, no biologist is going to have that, at least not the ones that are generating these sequencing datasets. And so the AlphaFold module is not a server call. It actually runs a simplified version of AlphaFold locally on your computer, and I wrote all of my gget modules so that they run on any laptop, so they have very minimal requirements for computational resources. So I just wanted to mention. Did
you realize how big of a project this was going to be when you started? Because maybe because I don't like programming particularly, this seems like an enormous task to me, but it probably also actually is.
No. I did not set out to build this at all.
Because it seems like you're almost like a software developer now with a biology background.
Certainly gget, I think, is becoming more of a software development challenge. Though, you still need a lot of biological understanding to even know which problems you need to solve. But I did not set out to build gget. It started because I was getting really annoyed and tired of having to constantly look up different genes and different gene IDs that are completely meaningless. They're just a bunch of letters and numbers manually on these websites. It just frustrated me so much. It was a huge, I thought, waste of time that I had to keep doing it. And I also was very error prone because you're manually copy pasting information. And so that's how I started building gget. And I built this first module, 2 modules, gget search and gget info. They basically do exactly that, just finding information about genes, combining information from different databases. And it just came up in a conversation with Lior, and he was like, you should make this public. Other people might use it. And we were like, yeah, maybe there will be like 2 or 3 other people who will find this useful. We'll make it public. We'll post about it. And then it went completely viral. And now it's become a huge thing. Yeah.
I mean, obviously, I only have hindsight bias now, but it does seem obvious that if there's something that replaces time-consuming and error-prone manual search automatic, people will like that.
Yeah, no, I did not realize how many people had this problem, and also that there wasn't really a good solution out there for this problem. And I think a huge part is that nobody who really had this very deep biological understanding, and also an understanding of these biologists, they obviously know how to code because they're analyzing this data, but they don't necessarily know how to make an API call. They don't necessarily know the intricacies of Bash and Python functions. And so I really wanted something very easy. 1 line of code, I think, is one of our ethos at gget, is every module solves a problem in 1 line of code in the simplest form. And then you can build from that. But yeah, that was really important to me. And it turns out other people had the same problem.
Yeah, but I guess it's a lucky coincidence in that sense that, as you said, because you have to have this, as you said, first about biological knowledge for what you even need to do and how you would like it to be done. But also, yes, that's also what I meant with like, it's a big software project where like, I know how to code. Like I can do my analysis, I can run in psychology, my stimuli, I can do that, but I don't want to have to deal with all the stuff you just mentioned. I've heard of these things, but I guess you found a really nice combination where I guess stuff that other people, they would not like one or the other, you like both of them.
Exactly. Yeah. I love it. I really enjoyed working on gget. I mean, like I said, I was building it for myself. So the first version, I was just, when I had time on the weekend, I was sitting at a cafe and just working on it. For me, it was also, I was still teaching myself how to code in Python. So it was a great problem to do that. Every once in a while, I visit some of the old modules and I'm like, oh God, I need to replace all of this code. This is terrible. We've done a pretty good job of that. We've made it a lot more efficient. But the first version worked and it was correct and it was super useful. And actually, I mean, I always tell this to young biologists who are like, why would I bother to learn how to code? There's all of these people out there who are so much better than me. And I always say that, they might write better code, but they don't necessarily know which problems to tackle. And you can write beautiful code. If it's tackling the wrong problem, then it totally doesn't matter. So. Yeah,
and I mean, I think the standard advice for like people who want to be entrepreneurs or that kind of thing is often take a problem you have and solve it.
Yes, that too. Yeah.
That's exactly what you did, right?
Yeah, exactly. It was just a problem I had. And I think I hope next time I will have more confidence to know that if I have this problem, probably other people do too. And it's probably worth solving.
I mean, obviously it's also lucky that you did solve a problem that lots of people wanted. There is also an alternate universe where you put the same amount and like no one really cares.
Yes. Yeah. No. Yeah. 100%. I would say luck was also involved. Yeah.
But okay, so I mean, you've talked about like the regular updating and checking whether it works and all that kind of stuff. So from what I understand, you're going to start a postdoc soon somewhere else. So what's the kind of plan there for maintaining gget? Is it just going to follow you now? This is your child. It's going to be with you for your life.
I haven't quite figured it out because, I mean, gget was supposed to be a side project. It is not my PhD project. My PhD project was on detecting viral sequences in RNA sequencing data. And gget is really a helping tool in all of my work, but maintaining it is technically a side project, which has just invaded a lot of my life. So I would love to get a more sustainable long-term environment where gget. So one of the things that I've been working on when I got funding from the Prototype Fund in Germany, one of the things I really worked on a lot was community building. And that worked quite well, which basically entails that I cleaned up the structure of gget and wrote very detailed instructions such that biologists and other experts can now, when they want a new module, they have a problem that I haven't solved yet, or maybe that I don't understand well enough, I can guide them to write it themselves. And so that is something that at least, and it happened already, we already have 4 or 5 community contributed modules, which is also great for them because they can now put it on their CV. They wrote code that is very widely used, and it's part of this very widely used coding environment. So that is something that I've been working on, and I would love to see that more. I would love to see more community involvement. Yeah, I set up a GitHub sponsorship page.
What is that?
Oh, it's basically you can get sponsors if people are fans of your work.
I'll give you money, basically, or
basically, yeah. It's
okay,
a buy me a coffee. I think it's a similar page. Exactly. Yeah, it's the exact same thing. So I set up one of those. It's always nice to get a little bit of financial support for your work because, I am just doing it in my free time, unpaid. If I'm being honest, I don't see myself stopping anytime soon. I feel a huge responsibility. Like whether or not I get financial support, I don't think I'm going to stop. I do feel a huge responsibility towards my users. There's a huge list in GitHub issues, there's a huge list of requests for new modules. So that is something that I need to be very careful of whether or not I have time for that. But whenever something breaks or there's a bug, that gets addressed immediately. So I do make sure that everything that is in the gget environment is correct and reliable. Because, I definitely feel that responsibility to my users.
It's good to see that you have the same, not particularly sophisticated negotiation strategy that I have of saying, hey, you can give me money for it, but we'll do it for free. I'll do it anyway, even if you don't pay me. But
if I'm being entirely honest, I would be very disappointed in myself if gget didn't work anymore in the future. So I have a Patreon for the podcast, but number one, I've basically never mentioned it, and number two, it's I'll do it anyway. It doesn't depend on that. I'm not sure I'm not sure it's the best negotiation strategy, but I don't know, it makes life easy. I think that I think we should get paid for the work we do. I think we're contributing to the scientific community, but I think part of why you have so many listeners and I have so many users is because we are doing it because we really care. So I think partly because we are going to do it anyways, it is so useful. So there's a bit of a...
and in your case also, more than in mine, you're also becoming maybe not indispensable part of the scientific community, but it is very much, it seems like it's a tool that people really use and makes people's life a lot easier. So I guess in some sense it is for you also some sort of, not job guarantee, but I would imagine it does help you quite a lot in getting
Absolutely.
a job, right?
Career-wise, it is really great to show that I'm able to produce something that people love and use and rely on.
How many people can say, if you hire me, you hire the person who's responsible for this thing that all of you use? Not exactly like that, but that's what you can say, right?
I've benefited a lot from gget's success. So absolutely.
But it's obviously still cool that you're doing it. But okay, so this is something I wasn't quite sure about. So your main research is still something not completely separate, but it's separate from gget, and that's just a part of what you did, and now it's more maintaining and.
I will say gget's success has tinted all of my PhD projects in that I think a lot more carefully about this niche that I try to be in between biology and bioinformatics. So I really try to make all of my projects, everything I do, very accessible. Like I recently published a paper on finding viral sequences in RNA sequencing, and it's a big paper with a lot of figures, but every single figure has something called a Google Colab notebook. I don't know if you're familiar with it.
Vaguely, but not really. I've seen it and used it once, but yeah, not really.
It's great. It's basically a Google Doc, but for Python code, and that it is an entirely contained coding environment, and you can just press play and the analysis runs. You don't need to install anything locally on your computer. And it's really nice because you can really see how this figure was produced, and you can even change things along the way and see how that affects the results. And so every figure of my paper has a notebook like that anybody can just run. All you need is a browser, and you can reproduce that figure. And then also, right, you can reuse my code if you want to make the same figure, but with your data. So yeah, in that sense, gget has become a huge part of my thesis, but I've actually done a lot of work outside of gget.
Yeah, I really like the whole, what you mentioned with, being able to share all the code. And I mean, I guess the data you use is public from what I understand. But yeah, it's basically, I'm always torn between, so I learned coding in MATLAB because that's what most cognitive neuroscientists use. And I just don't like coding. So I've wanted to switch to Python for ages, but it's always like, it's the part I like the least. The problem with MATLAB is really, I mean, in cognitive or computational neuroscience with humans, like lots of people use it. But there's this point where you share all of it and people are just like, yeah, I don't use MATLAB. I can't read it. So it's technically, it's all open. But if lots of people can't use it, then how open is it really? So there's a bit of but I just can't bring myself quite to learn a whole new programming language.
I think Python in general, obviously, I think it has the most users of any programming language, at least of the ones we use in biology and in my field of biology. But that's why Google Colabs, I think, are so powerful, because you literally just hit run and you don't... I mean, MATLAB is also a problem, right? Because it's not free. So is it really open source if you need to pay for a program to actually then run the code?
Yeah, exactly. It's open in the sense that anyone can evaluate what I did. But yeah, it's not... I mean, to be fair, like no, you're right. Just my aversion to programming is strong enough.
I can recommend Python. I love it.
I've decided to switch in the beginning of my PhD and the beginning of my postdoc, and each time I was just like, but I know how the other works. And then it's like learning how to walk again. Where, like, of course, you know in principle how to do it, but every step is what's that function called and all that kind of stuff.
I had to. So for my main PhD project, I had to write a lot of code in C++, and that was so painful. So I feel you. Yeah,
I mean, I already have it with R because it seems like in lots of stuff I do, you don't get around R. But
that's
I mean, I think one day I'll switch, but by that time, I don't know, people probably not be using Python anymore. It's probably by the time I switch, it's not going to be cool anymore. It's going to be some whole new programming language none of us know about.
Is up and coming, and a lot of people that I know swear on Rust. So maybe you can get a head start.
I'll get a head start on that. Yeah, or people don't use that, and I've got a head start on nothing. Yeah, we'll see. For now, I've got enough to learn while keeping the programming a constant.
I mean, that's the problem, right? If you're the one generating data, if you're the experimenter, it's incredibly difficult to also be able to learn these very technical skills. I mean, I spent my entire PhD learning this full time. I don't know how anybody expects somebody who does wet lab work full time to also have the same level of knowledge.
Yeah. Okay, but your postdoc, what are you doing there? Because that's going to start, what did you say, like 2 months or something?
September, yeah. So my postdoc, I'm joining a lab at the Broad Institute in Cambridge, and I'm joining the lab of Professor Pardis Sabeti, and she is just an incredible person. I think it's fair to say that in part thanks to her, we never had to worry about Ebola outside of Africa. So she has done a lot of work on Ebola virus during the outbreak. Her lab has done a lot of sequencing of genomes, a lot of rapid test development to very quickly react to new pandemics and keep them contained. So like I said, my latest and big project was on detecting viral sequences in sequencing data, specifically new viruses. So even trying to recognize viruses that we've never seen before. Because with RNA sequencing data, you sequence all the RNA. But then the way it's analyzed is you compare it to a reference, and that's how you figure out what is what. So anything that is not in the reference just gets thrown out. And so I try to focus on that part that is thrown out. And I built an algorithm that can basically learn what looks like it's a viral sequence and then pull out those reads. And then also try to figure out which virus it came from, whether it's a new virus or something we've seen before. And then I can also see so I'm mostly working with human cells, I can see how the human cell responds to the virus being there. And so my preprint is out describing all of that, putting in
I'll put it in the description.
Yeah, wonderful. It's under review right now. But that was just the first step, because this was, again, an entirely computational. I think I have potentially identified some new virus-like sequences that have an effect on cells that they are infecting or that they are present in. But there's not much else I can do with that right now. And so I want to continue that work in her lab. And basically what I'm going to be doing is analyze a lot of really amazing data. I mean, the Broad Institute, they are the epicenter for genetics, genomics, and sequencing data. And so I'm going to be hunting for viruses, and I'm going to see if I can actually extract them, trace them. I've been also doing a lot of work with cancer. So we know that a lot of cancers have viral causes. So yeah, I'm trying to just see if there's some correlations that perhaps we have missed. And obviously, if a cancer has a viral cause and you don't know it, like that might be a relatively straightforward treatment route, and also early detection and early treatment route that we're not aware of.
Virus Hunter is a pretty cool title.
Yep. Yeah. It's very contemporary as well.
Yeah.
I do want to say that any viruses that I would hunt would already be out there in humans. So
yeah, you're not creating them.
Yeah, I'm not creating them.
Not yet. Yeah.
Yeah, I would love to continue that work and just really apply it at honestly a global scale and see if we can. So Pardis and her lab, they are extremely passionate about global surveillance of viruses, not accepting that, oh, I just have something, the flu or this virus, maybe, I don't care. Just not accepting that anymore and just really getting better at keeping track of what viruses are doing, where they are, how they're spreading. And so I'm super excited to be part of that.
I mean, that also sounds like quite an administrative task, right? Because you have to coordinate all the different places and collect all the things centrally or something like that.
Absolutely, yeah. So they have a lot of collaborations with local institutions in Africa. And they also work with the CDC and the US government. And one of the things I'm really looking forward to learning from them is, how do you do all of that? How does that look? How do you even set up a global viral surveillance system? Where do you even start? So,
it's funny. On your website, you mentioned that you do a postdoc there soon. So I had a brief look at the website, and I was like, this is not a lab, this is an institute. Because it's like she has about 10 people working in administrative stuff. And I was like, what are they doing there? But now that you've explained it a bit more, it makes a bit more sense why there's so much, not just, I mean, there's lots and lots like science, like postdocs, that kind of stuff, and PhD students, but like, now I understand why there's so many of those people too.
Yeah. Her lab is, a huge part of her lab is basically run like a company, and they do very immediate reactions to outbreaks and very rapid test development. And so she has that infrastructure to do that at a relatively large scale in her lab. Yeah.
It all makes sense now. Yeah. Okay, so let's talk about a little journal club you did a few years ago. What happened?
Yeah, okay, so this is completely unrelated to any of my work. During my rotations at Caltech, one of my rotations was in an insect behavior lab. And for a journal club in that lab, I was assigned 2 very fundamental papers to the field of the honeybee waggle dance. So what is the honeybee waggle dance? Sorry, that was your part.
You can do both. Yeah, you can interview yourself if you want. I'm happy to just listen. I'll edit it and publish it. You can do the rest. Yeah.
No, so
what is the honeybee waggle dance?
Yes, good question.
Thank you.
So we've known for a long time that the honeybees dance to communicate the location of food. And I should disclose again, insect behavior is absolutely not my expertise. So I'm just reporting on my understanding of it. But we've known for a long time that honeybees use the waggle dance to communicate the direction and distance of food sources to other honeybees so that they can then find the food source as well. And so the papers that I was reading were trying to figure out exactly how honeybees track the distance. Do they remember certain monuments along the way? Do they track their energy consumptions? Do they track their wing beats? There were a lot of different theories on how honeybees might do this. And the 2 papers that I was reading, and this was the fundamental work of the first author on those papers, and it was very fundamental to the field, is that he found that honeybees do that based on visual cues. So basically visual motion is how they track distance flown. And then he also tried to make in these 2 papers a very specific calibration of this many seconds of dance mean this many kilometers in distance or meters in distance to the food.
So just it's funny that like I've done lots of episodes on the neural basis of spatial navigation, and only just now it occurred to me that this kind of also fits into that. But yeah, so we do usually just like in rodents or in mammals. But yeah, I guess that actually that's pretty much the same question. Yeah.
I mean, it's super interesting, and I quite enjoyed reading about the experimental designs actually. But well, okay, why am I telling you about honeybee dance as a computational geneticist? Basically what happened is that I was reading these papers. I had no idea about the honeybee waggle dance, so I started reading other papers by the same author. I realized that I was looking at the same data over and over again. Basically, as I was trying to really understand what was going on, I was also trying to redo some calculations. So the specific calibration number, I was trying to understand it by actually redoing the analysis, and I just kept getting a different number. So this is really how it all started, and then from there I started very carefully analyzing the data, and I found that several of the figures were showing identical data, but for different experiments with different numbers of replicates.
Yeah. Okay, so I mean, so you found some. Yeah. So basically, what did you do? This is. Yeah. Not what you would expect to find? Yeah,
I was pretty concerned, really concerned about those findings, and I had no idea what to do. So first, I presented my findings at the journal club because I also wanted the feedback from actual experts in the field. And that is really where this most shocking response happened that stuck with me for my entire PhD afterwards, which is that nobody cared. The response was just, oh, yeah, that sucks. Huh. Oh, well. And I reached out specifically to the professor of the lab, and he basically said he didn't know what to do, which is a little bit better than not caring entirely, but also he didn't try very hard. And also, you're a tenured professor at Caltech. What do you mean you don't know what to do about the data integrity issue? So I reached out to another professor at Caltech. This is the person who taught the ethics class that year to all the first year students. So I just had to listen to him for a month, preach to us about data ethics, and it's so important. And if you make any mistake, you're horrible. And he just basically told me, oh yeah, this happens. Science is messy. Do you really want to waste your time on this? And I got really frustrated. And at the time, I mean, I was a first year student. I wasn't going to cause any trouble on my own. Well, I did a little bit because I did tweet about it. And then I got some help from Dr. Elizabeth Big, who's obviously amazing. She's doing some really, really important work in the field of data integrity. And so she helped me find the papers on Papier and at least flag the issues that I found on Papier, where they have remained ignored until pretty much this month.
Yeah. So there's an entire episode with Elizabeth that I'll also link in the show notes where we talk about awesome. All sorts of stuff. I mean, related to this particular. Yeah, I mean, so maybe just to, I guess we've set the scene a little bit, to, motivate it a little bit why we're talking about this is that, I mean, so I've had basically 2 episodes before about this kind of topic. The first with Joe Hilgard and the second with Elizabeth Big. And basically the first one I had with Joe in large part was because I also found some irregularities in a paper where I basically thought 2 figures were mutually exclusive. Like there was a specific set of findings where wait a minute, these can't both be correct. And I just had I presented it in our lab meeting just in the lab. And basically everyone's like, yeah, that doesn't look good. But just no one knew what to do, right? You're not trained to, if this happens, then this is what you do, right? And so one of the reasons I invited Joe to talk about it is because I was just what do I, Joe, what do I do? Yeah. And I mean, interestingly, he is, in particular, I interviewed him at the time because he found this ridiculous fraud case in his own field, which is how he got into it, because someone in China reported studies with thousands of people where usually you just have 100 or something. So it messed with his entire meta-analysis he was running. Anyway, so we talked about that. And then I asked him also what I should do. And he, as someone who has spent lots of time reporting these things and getting papers retracted and that kind of thing, basically also told me you know what, it's probably not worth it. It takes a lot of time. You have no idea how the people are going to react. Mm-hmm. And yeah, it's potentially a lot of time that just goes to waste. And then I didn't do anything during my PhD also because you have other stuff to do. But then when I talked to Elizabeth a few months ago, I actually, because this was between my PhD and my postdoc, I had a bit of time. So I actually wrote it up, contacted the original people. And it looks like they are the best case scenario, where they basically said, they responded within 2 days. They said, yeah, this basically we're taking it seriously. Awesome. Mm-hmm. And they already contacted the journal to ask what to do exactly. Yeah. I mean, so yeah, it's funny because Elizabeth, I asked her and Joe what to do, and both of them were I mean, you almost don't need to bother contacting the authors because they'd never respond and that kind of stuff. Anyway, sorry, I didn't want to interject that much, but the point was that, I mean, this is a very unusual, I mean, it's not an unusual, it's not necessarily a rare situation, but it's a situation that it seems like the entire system isn't exactly prepared for. And we're probably not the only people who have found something like this.
No, I mean, I have gotten so many messages from especially early career scientists telling me that the exact same thing happened to them.
Yeah. And so I guess the idea is to share your story and to some extent continue my story in little bits over the years now. In a year, I'll do another episode then probably and finish it or something like that. Yeah, to just give people some idea of what to do and some sort of suggestion or guidance or something like that. So maybe going back to your story. So so you did nothing. Okay, cool story.
Yeah, I did nothing. The story does continue. I still haven't quite decided yet if it's a happy end or not. So I don't even know how much it's really helpful in giving advice, or if it again.
Well, it's at least showing examples, because it seems most people haven't even heard of examples, right? It's basically, it seems like I have the examples of Joe and Elizabeth who've done this a lot, especially in Elizabeth's case. Yeah. But it's also very different if you are, I mean, so if you're, in your case, I guess, one interesting commonality to me is, and maybe you can tell me whether this is a coincidence or not, is that both of us have, in my case right now, I've only mailed them and it's behind closed doors for now, but there will be some sort of public statement at some point soon. Yours is then public, but both of ours are cases where the field or the papers we're criticizing were from a field that was adjacent but not our own.
Yes.
And I have to admit one thing, I contacted them myself. I just wrote them an email by myself, and it's scary. Like, you write this report and you're basically saying, in my case, it's three, seems like fairly senior, well-respected professors, and you're going like, hey, you three professors, I'm a PhD student. I think you're wrong.
Yep. No, it absolutely. And I think having it be in an adjacent field makes it a lot easier, because you already mentioned you never know how the authors are going to react. And I mean, that is something that I was told a lot, is that, do I really want to jeopardize my career? I think in the end, that was really bad advice. No young scientist should ever hear that in a response of them doing their duty as a scientist and ensuring data integrity.
I mean, but don't you think it's also, I mean, you don't know.
A lot of, right? That's true. Yeah, certainly if it's in your field, unfortunately, even if you're right, it can really hurt, especially younger scientists.
Yeah.
They anger the wrong person.
Yeah, exactly. So, I mean, in my case, it was basically, I mean, it's interesting because in my case, I didn't have access to the data. I just found some irregularities and where I thought there was stuff that was mutually exclusive. So I got an email from them like a week ago where they said, like, it took them a while to get the data, which makes sense. It was like a while ago that they published it and that kind of stuff. But they basically said, like, okay, we got hold of the data again, we reanalyzed, and we think your initial point is actually not correct. There's some other stuff, and we are going to issue a correction. Like, there is some stuff, like some of the stuff you correct is correct, but like the big point you had is we think that's actually true the way we have it in the paper. And so I'm still really surprised by that, and I'm looking forward to getting the data and seeing how it can be true. But, you know, in my case, I basically just showed it to some people, the draft I wrote, what do you think of it, that kind of stuff. And then I sent it thinking, like, they're not from my field. What's the worst that can happen? That's basically it, right? Yeah. But yeah, if it's exactly my field.
It's scary. It's very scary. You also mentioned a great point. I'm skipping ahead a little bit here, but yeah, so I mean, my story ends with eventually me and my current advisor, Lior, we wrote a report and we figured out that there's just nowhere to publish it. And there's certainly nowhere to publish it with a peer review. We really wanted a second pair of eyes on this report as well. We're very confident in our conclusions. Obviously, now we've had a lot of peer review because it has gotten a lot of attention, which is great that this is exactly what we wanted.
But have you gotten actually good peer review, or is it more like, you know, a lot of noise and people react to it in more like a social commentary style rather than actually, you know, there's a difference between you like knowing you can anonymously give feedback on this thing and spending time with it versus seeing something tweeted and then checking over it quickly.
Yeah. None of the issues we brought forward have been clarified. I'm not entirely surprised by that because the issues that we found are just not really debatable in that way. The data is not very similar. It is identical, and it is for different experiments. So I think in that way, there's just no way in which you can be like, oh yeah, we looked at the underlying data and it actually looked at that. It's like that. No. But we did get some helpful comments initially, actually, when we submitted to the Journal of Experimental Biology. They rejected the manuscript, but one of the scientists working for them contacted us separately and said, off the record, here are some suggestions that I found in the report, and that actually was super useful just in the phrasing and how to explain what we did, because obviously we know very well what we did. So sometimes it's just really nice to have a second pair of eyes, especially in that field. And so that was super helpful.
So what's the current situation? I mean, I know you published the preprint. It got some attention, including from the original author. So I think he said he was going to respond.
Yeah, he promised in detailed response. Yeah, you were talking about the best case scenario. I think we have one of the worst case scenarios, unfortunately. It was quite bizarre because, for example, the Journal of Experimental Biology, they published 4 of the problematic papers, 2 of them with very obvious data duplications. And the journal actually posted a correction in which they list all of the duplications. So they say themselves, this data is duplicated here for this experiment. with this number of replicates, and then this data is duplicated here, and it's a whole list. It just goes on and on. But then they conclude with the author takes full responsibility, the data is not available anymore, but the conclusions are not changed. The conclusions that are drawn from the data, which we have just established, at least one of the two sets is obviously wrong because it can't be identical. And so that is really bizarre to me. So I think the other journals are still investigating. I know from Science covered a story, and I know that their internal investigation is ongoing. So I'm looking forward to see how other journals are reacting.
Because one of the papers was also published in Science, right?
Yes, exactly. Yeah. I mean, I think it's like 7 or 8 different journals that are impacted by these papers. Yeah, I'm very disappointed in the handling. I mean, I'm now only talking about the journal responses, but yeah, the Journal of Experimental Biology, it also looks from the responses of both the author and the journal that they were aware of these issues a year ago. The author himself has commented on our blog post that they were contacted by the journal a year ago. That wasn't us. That was way before we even started writing our report. And so it sounds like both him and the journal just decided to not mention it, not publish any sort of response, or just making the readers aware that this is happening in these papers until we published our report. And then a month later, it sounds like the journal published the responses that the author gave a year before to only those issues.
Right. Oh, so it isn't actually... Okay, so you actually, so just to get the timeline correct then, so you published the preprint, well, but you did contact the journals first, right? So basically the question is the comments that the journal officially made, is that in response to your stuff, or is that independent of that?
Yes and no. So let's just do the whole timeline. So I first discovered the issues in 2020, in the first year of my PhD. That resulted in those 2 peer comments, which were completely ignored by the author and the journal. And then in the last year of my PhD, so that was like October last year, this came up again in a discussion with Lior, actually because we were talking about what I wanted to do after my PhD. And I was still so frustrated by this thing that happened in my first year that I was like, I don't know if I believe in science anymore. Like that just like goes against everything I've been taught. And Lior is the kind of person who will not let something like that slide, which I really admire about him. I think we need more people in science like him. But so we wrote our report. We sent it to the Journal of Experimental Biology in March because several of their papers are affected, because we wanted a peer review process, and we thought that they might be interested in publishing this kind of report on work that has been published mostly in their journal. They rejected the preprint, they rejected the manuscript without sending it out to peer review, said that they would start an internal investigation.
So just briefly, so it was just wrong type of publication basically, or what was their...
They said that they didn't want to publish anything that includes criticisms of works that are published in other journals. And also generally, I mean, they really just wanted to focus on the research finding on this calibration number and not talk about any of the duplicated images. And we said the whole point is that this happened over and over again. It needs to stay together. If you look at any one piece of the story, it's not meaningful. It's the whole of the sum that is greater.
Okay, sorry, I interrupted you. So they started an internal investigation.
Yeah, so they rejected the manuscript, but told us that they started an internal investigation. Nothing happened. We said, okay, whatever, we'll publish our manuscript on the archive so that at least it's out there and we just know that this happened. And then a month after we published our archive manuscript, they published their expressions of concerns. But now, based on what the author has commented on our blog post, I'm wondering whether they just posted the responses that they actually got a year ago. And I mean, this is completely speculation, but my guess is that they knew those responses were really bad. And so they just rather not talk about it, hope that nothing else would happen. And then when we published our manuscript, we basically, through that, forced them to acknowledge the problem. And that's really disappointing, right? The fact that in this case, we had to go public like that to get any sort of response from even just one of the journals and the author, that's really disappointing and scary.
Yeah. I mean, as you said, like some of this was maybe a little bit of speculation, but one question I had is basically about contacting the author. So in my case, I was like, okay, I mean, first of all, my case seemed it seemed less severe than what you found. And it seemed like there was a solid chance there was just some error in the analysis and it wasn't done that carefully. That's basically, it was very possible. And also I didn't read any other papers by the people who did any of that. I had just looked at the one paper. But in my case, so basically, I initially thought, so I asked, I think, Elizabeth, what should I do? And I think she said, basically, just contact the journal, put a comment on pub here, basically what you did, right? But then for me, I don't know, it didn't quite sit right to not let the people know before I did this. So I sent them all an email, included them all in it. And then, I say it's the best case scenario. So far, nothing has actually happened. They just say, I mean, I don't want to be too distrustful, but I haven't actually gotten the data or anything like that, right? We're still a bit behind some of the things. But if, let's say in my case, it turns out that their initial reanalysis is correct, then I'm quite glad that I went to them first and they can correct it. Yeah, I'm curious. It seems like you didn't do that, right?
Mm-hmm. That's correct. Yeah, we didn't contact the author directly. Part of that was because of the nature of the problems that we found. There was no really debating the issues that we were finding. That said, obviously, like I said, I very much encourage discussion on the preprint that we put out. We really tried to describe in detail what we found and where, and any analysis that we did do is there and is reproducible. But yeah, I wanted to be able to stay objective in this and giving the nature of the issues. I didn't think I could do that. I also I didn't see how meeting the author and talking to the author would add anything to it. And in addition to that, those PubPeer comments had been sitting on PubPeer for 4 years. And Srinivas, the author himself, has confirmed that he has known about the issues, even if he didn't see the PubPeer comments, and PubPeer does send authors an email when the comments appear on their papers. But even if he didn't see those, he has admitted himself that he has known about these issues for a year. And so he has had a lot of time to address these issues, to do the right thing, and he didn't. And so I think, I don't see how us contacting the author.
Yeah, right. I see. It's a different kind of problem in that sense. Yeah, if you think you've found systematic problems across several publications compared to me having looked at one paper and going, okay, I don't think this is right. Yeah, that's obviously okay. Yeah, I mean, I almost don't want to say this because it might give away some stuff. But one interesting thing that Elizabeth said is that you can basically, from the response of the people, tell what happened. So she said basically, if the people who actually made an honest mistake will respond immediately and be very friendly and grateful to you, which is pretty much what the people did in my case. And the people who maybe did something more serious tend to start attacking the people and questioning why they even did it in the first place. And there was a little bit of that in the comment on the blog post.
Oh, very much. Yeah, no, I mean, there has been zero addressing of the issues that we found and a lot of personal attacks on Lior. I mean, he literally called us unprofessional. And if you read our manuscript, we're not even mentioning any one person by name. We are just correctly citing papers and listing the issues we've found in each paper.
But I mean, to be fair, it is... I know you don't...
I mean, obviously, it happens that most of the papers have a common first and last author, but we are just listing the papers and listing the problems that we found. So, I had been asked how I would respond to that, and it's really difficult because I honestly don't know what we did that was unprofessional or unscientific. I think we wrote a scientific paper. We made it very transparent, very clear. We invited comments. This is why we wrote the blog post, is we wanted a platform where people could respond. And we are definitely inviting experts, including the author, to comment on our technical analyses. But that has not happened at all.
It's really weird. Like I can kind of understand why people feel attacked by these kind of things, even if it's done in a completely professional way. Like I think it's a completely natural response, but I think this is also part of the bigger problem, right? Where it's just somehow it's never done, right? It just happens so rarely. And I mean, there might be people like Elizabeth who've stepped aside basically out of academia itself and do this as their thing, and who seem to have a thick skin and don't mind being attacked by people.
And look what she has been through. I mean, she gets attacked all the time, and it's completely unprofessional.
Yeah, but it's so weird how, I think it's just because it happens so rarely. When someone does it, I guess it's often seen like a personal attack or something.
Yeah, and honestly, I think that's part of the problem. I think that retractions shouldn't have as much of a stigma as they do. Like, everybody makes mistakes, or sometimes we just simply get it wrong. A huge part of science is just sometimes you just get it wrong, just because we are totally fine with getting it wrong until the paper is published. Then all of a sudden it has to be holy.
Yeah.
And it's really weird. Like, we should be able to, what we did was peer review. And for some reason, the Journal of Experimental Biology just didn't accept it as peer review because it happened after the paper was published. And I think that is very detrimental to correcting scientific literature. And I think part of that is the fault of us as scientists, as a scientific community. I think, obviously, again, our issues are of a very difficult nature, but there are a lot of restrictions where the science just doesn't hold anymore based on new evidence, and then it should be totally fine to retract the record.
Yeah, I like your comment about peer review, because I've had a few situations where I'm always surprised that people think peer review is only a thing that happens when you submit it to a journal and you get invited by the journal. Everything else is somehow not counted as peer review.
No,
which is funny to me.
Comments on papier are peer review. Comments on bioRxiv are really valuable peer review.
Yeah. I mean, I've mentioned this example once on the podcast before, when we were reviewing a paper and they basically made a claim that no one had done something. We said, well, there's 3 preprints on this. It doesn't really matter, like, it's not, but just mention them. And they basically said, like, we don't cite preprints. Like, we want to go through peer review. And it's like, you are a peer. Like, you can read it and do your own peer review. Like, you can do it, I think it's a huge problem. Okay. Yeah, so I guess one kind of thing that's, not unfortunate, that's maybe the wrong word, but we don't have the official response by the author yet. Who knows what he's going to say? So this isn't about, I hope our discussion isn't about this particular person or even the honeybee papers, but it's, as we said earlier, I hope it's more that other people who find this have some sort of something to grasp onto. Or some examples. I'm curious, what would you do it again? What would you recommend yourself a few years ago? Or
I think it's a little bit early to say that. I think that going in... So I want to just very clearly say my goal here was to correct the scientific record. There were very obvious problems, and I felt strongly that people should know about them. And that was my only goal. And I think that I got really lucky in that I was giving a platform via Twitter, via Lior's blog, to be able to get that message out and inform the readership. I mean, our findings were very widely shared within the community of insect behavior and etymology, and that's great. I'm very happy to see that. That was my goal, is to let them know. So I think in that sense, my goal has been achieved. I think while doing that, we realized that I shouldn't have to have gotten lucky to achieve that goal.
Right.
There should have been a platform to do that, and there should be some incentive for young scientists to to talk about their findings. Again, I've had so many people reach out to me with similar stories, and who knows what they found? Who knows which findings we're building on that at least several people in the world already know are not actually correct. Like, that's bad for science everywhere. Like, that is lethal. So literally lives depend on science, and on science being, especially foundational papers like these, we need to be able to build on them. So in that sense, I've achieved my goal. I'm very happy about that. I hope that moving forward, this issue of there not being any defined way of getting word out like this will get some attention, and maybe we can even work on solutions to it. Leo and I write about the Journal of Scientific Integrity. Now, if we can get that and then also give it the same status as Nature, that would be great. But yeah, this kind of work, it requires, it's really difficult work. It requires a lot of very careful analysis. And right now there's just no incentive to do that. So I would love to see that be solved in some way in the future. And if this was a step in that direction, then yeah, absolutely, I would do it again.
Okay. Yeah. It's such a difficult, I mean, for anyone who's in that kind of situation, I find I still don't have a good answer to what to do, because I think in our case, we both maybe got lucky in different ways to some extent, assuming that my case continues the way. I mean, they even said, like, we told the journal we're going to write a correction and we want to acknowledge you for letting us know about this, right? So, like, for me, it's great. I can put this on my CV and say, I was acknowledged for this in the corrections and whatever, because it was a lot of work, right? Because, like, you do check your work 5 times before you tell people that you think they're wrong.
Course. Yeah.
And but the probability that someone does the same and is just told to fuck off or ignored is, and then you've just spent lots of times and someone just shouted at you. Yeah, great.
Yes, exactly. I mean, I spent weeks on this initial analysis.
Yours was much more extensive.
So the preprint then, but I meant even like the initial, just a few issues raised on Papier. I mean, I was a first-year student. I really went through those papers over and over again, because I really wanted to make sure that it wasn't just me making a mistake, of course. So, and that, all of that work, just for it to be completely ignored for 4 years, right? And
I guess you did have that case, actually, But so what's the status now? You said you couldn't find a journal. What's the...
So actually now that there's been so much attention for the issues that we raised, we've had several journals either interact on Twitter or actually reach out to us. So, for example, apparently there is a journal of trial and error. I have not really looked into it in detail. Yeah, it's a relatively young journal. I think the founder is literally a grad student.
Okay.
I really hope. I hope I'm not getting this wrong. I have not spent that much time on this journal, but there are options. So we are thinking about it. Again, our goal was to get the word out and get peer review. So we've achieved those goals. So I don't really see how getting it into a journal adds much, except that maybe it will appease some like random people who are like, anything that's in a preprint doesn't exist.
And I guess if you have it a preprint, then it will be findable. But I guess it's still, it makes a difference if you get the actual. I guess you would have to get the actual journals to do something about it for it to like permanently be visible for people who search the paper or something like that.
Well, actually, so I mean, if you put out a preprint, it has a DOI, so it is a permanent record.
But I mean, like if let's say I want to learn about honeybees, and or bumblebees, but let's say I want to learn about honeybees, and I find those papers. And I mean, there's always a problem, right? Like, how do you find out that there are corrections or retractions or anything like that?
Of course.
But having it on the actual journal is, I guess, the biggest step.
Exactly. Yeah, I am really looking forward to hopefully seeing some more responses from the journals. But that will take.
Yes, especially because, I mean, now there was a lot of attention, but like, give it a year. How many people who are then starting off as grad students will know about this, right? I mean, it will...
Yes. Yeah, exactly. Yeah. No, I really hope to see And I think that if the responses from the authors remain the same, then a retraction is absolutely the correct way to go, because we simply don't have any data supporting the conclusions made. So I would love to see more responses from the journalists, and I'm really looking forward to them.
Okay. Maybe final question on this point. How has this changed your view now on science and academia? Is it too early to tell still? Or
Well, okay. I think that honestly what changed my view the most was Lior telling me that I can join academia and I can be, just because academia is not perfect, I don't have to act like a bad actor. So Lior was like, you can absolutely continue your path in academia and we can just write about this. We can just get the word out. We'll just do it. And you don't have to be like, oh, I want to be in academia, so I have to be quiet about issues of scientific integrity. Yeah, so that was very inspiring to me.
He basically did the old Gandhi, be the change you want to see in the world, basically.
Yeah. Yeah, so we'll see how it works out long term. But I think, to just have a very senior but supportive and optimistic mentor like that, who's not just, just keep your head down, shut up. That was very motivating and important to me. And that really made the biggest impact on how I see science, in that Lior can't be the only moral scientist. I've worked with a lot of great scientists, so I know that he isn't. And so, and then the responses that we got, especially on Twitter from scientists, were a lot of concern, a lot of care. And so that was also really amazing to see. And that restored a lot of my hope for science, I would say. So.
Okay. Yes, at the end of each episode, I have 3 recurring questions that I ask each guest. The first is, what's a book or paper you think more people should read? Famous, not famous, old, new, whatever. Just something you think people should read.
Well, actually, I was thinking about that. And if you don't mind, do you mind if I instead recommend a book instead of a journal article? A
book or paper. I mean, it can...
Oh, book or paper. Oh, yeah. Awesome. Perfect. Yes.
I hope you're going to recommend now, what's it, Von den Bienen by Frisch?
I think that's a great book. Yeah.
I haven't read it. Which is funny. Yeah. Anyway, sorry.
No, I've been thinking about the question, and I wanted to advertise reading books just for fun. So I feel like I've seen it a lot in my colleagues, especially. When they read a book, it has to fulfill some sort of function. Either it's like an educational book or it's classic literature, which is great, but it doesn't quite suck you in and give you back energy in the way that your favorite Netflix show does. And so I'm a huge fan of fantasy books. And so...
Twilight it is.
Twilight it is. I did.
Is that fantasy?
That was a long time ago.
Vampires, that counts.
No. But yeah, so one of the series that I read recently that is a bit more modern fantasy, not your typical dragons and fairies, but I thought it was a really nice series, was Rivers of London by Ben Aronovich. And it really had this like binge watching effect. You just don't want to put it down in the way that you just want to binge watch a series. And I feel like for some reason people are like, they don't have that for books. Like nobody is like, what am I going to learn from this Netflix series? But for some reason, there's a bit of a stigma against reading books just for fun, or at least that's what I've observed. So I wanted to advertise just a totally fun book. I hope that's okay.
It's your recommendation. It's not mine.
That's my recommendation.
That's totally fine.
Yeah.
Okay. As I'll put that in the description, everything we talked about in the description. Second question is something you wish you'd learned sooner. It can be personal, professional, whatever you want, but if you'd learned that maybe a little bit sooner, then it would have helped.
I think that in the beginning of my scientific career, I was very worried about doing science and just generally acting exactly like all the people around me did. And I was very afraid of speaking up, even just asking questions at a talk and things like that. Because I was just so worried that I would say something stupid or get something wrong. And so I think I wish I'd known earlier that it pays off to be present and be bold. And it's totally fine. Sometimes you get things wrong. It doesn't always work out. I think at the end of the day, you gain so much confidence and you feel so much more comfortable that you are actually able to do better work. So I wish I had known that earlier, that you can be professional and an excellent scientist and still also just be yourself. Probably.
How does one, like, is it just trying it and step by step doing more of it? Or how do you go about doing that and learning it and, like, applying it?
Yeah, I think having had a very supportive mentor in that, like I had in Lior, was very helpful. Yeah, I would definitely say just try it. I think generally being bold pays off. So, yeah.
Okay.
I wish I would have known that earlier. Yeah.
Okay. And then, usually I ask, So the third question is advice for PhD students or postdoc on that kind of transition period. I don't know, this is the first time now that I've asked this question for someone who's exactly on that transition period. So I don't know whether you want to take it exactly like that, or maybe you think you're still too much in that and you can pick any other kind of academic stage if you want to, but whatever you want.
Yeah, I'm literally in the middle of that transition period. So I would maybe give the advice to not take on a huge defrauding case in the middle of that transition period.
So it's funny because I also did, mine isn't that big as yours, but I also did it between. Yeah.
On the other hand, maybe this is the time period.
When else
you have the headspace, exactly, take in something else. No, but I think the most important advice I would give beginning PhD students is choose your advisor's character very carefully. And I think I really underestimated how much more important that is than their project. So at least for me, especially because I was willing to pretty much do any sort of scientific project and get excited about anything. Maybe that's a bit of a different situation for other people. But from my experience, from my own PhD and what I've also seen in other PhDs, is that what is way more important than the research that you're actually doing in your PhD is what sort of mentorship are you getting during it. And that can really make or break your PhD, I think.
Yeah, I agree. It's
maybe that's a bit standard advice. But.
I think so. Most of this, something I wish I'd learned sooner. It seems like there's about usually it's one of 10 answers. It tends to be, but I think they're worth repeating. And there's so many people I know or have heard of at least, who start a PhD having basically never met their supervisor before. Especially sometimes in Germany, you have this kind of where actually the supervisor is funding, you apply to them, and they've met them for 10 minutes. And then they go there, and maybe they've met some people from the lab, but maybe they haven't even done that. And then they go there, and everyone it's like the entire house is on fire basically because everyone hates each other. And I think it's worth repeating that. Yeah, make sure you're spending time with the right people for the next few years. And we say that, but honestly, it's easier said than done because at the end of the day, right? How much time do you really get to spend with your supervisor before you start your PhD? But at least you can ask about. You can meet the lab, meet the people. That's usually, to be honest, I think that's how you usually find out about the character of the supervisor is by the supervisor saying, please meet the people in my lab.
I agree.
Right?
Yeah.
Because if they don't do that, then that's already a bit of a, interesting. I
agree. Yeah. Yeah, I agree.
Okay.
Yeah.
Cool. Well, thank you very much. Exactly 2 hours. It'll be a bit under 2 hours once I meditate.
Yeah. We covered a lot of topics, so I hope I wasn't rambling too much, but
I don't think so.
Just really cool. Thanks.
Yeah, thanks. Actually, I have, this is basically a cut, but before I stop recording, I thought there's one question I thought I wanted to ask about Catalan. and Catalonia, which is a bit specific. So I think I might just include it at the end of this as a fun little afterthought, which is basically, so I have a very specific thing I'd like to do, and maybe Catalan plays a role in that or maybe not. And so as someone who has a background in that, maybe you have some sort of interesting insight into it, which is basically like so I want to do this postdoc for a few years. And then, I actually want to do postdoc for a while. So I want to do another postdoc after that because I just want to do science and learn stuff and not have like admin and that kind of stuff to do. But between the two postdocs, one thing I might do, which I think is like a little dream project I might do, is spend 6 months to a year going to a place I don't speak the language and then just like fully immersing myself, like learning the language for like several hours a day. But by the end, I have this whole like really be part of the community and learn the language and everything. And so I basically have this there's two thoughts I have on this. One is I want to learn the classic, like I want to learn a language that's obvious, like that it's very widely usable. Like Italian is, I guess, mainly in Italy, but Spanish would be an obvious one where if you speak Spanish, then you can speak to an entire continent. And there's a kind of obvious utility in learning these big languages. However, part of me also thinks that it would be super cool to learn a kind of weird language. And weird defined as in you only really use it in one area. I actually got the idea from Basque, because I thought when I was like,
that is a really
exactly,
that is a really weird language.
Exactly. It's if people are still listening, then Basque is a language isolate. It's literally not related to any other language on the planet, current or dead. They can't find any connection to anything, and so that might be a step too far because like you have, I'm assuming they have a lot of Spanish words in it just because a lot of the Spanish influences, but like the language itself, you'd have to learn every single word from scratch. There's no starting point. But anyway, so I was wondering, like, for example, Catalan might be like an in-between. It's like a unique small language that is, I didn't look this up, but I'm assuming it's a Romance language. So
it's a Latin language.
Yeah, exactly.
It's between Spanish and Italian.
Okay, so I don't speak either of those languages, but they're intuitive to me because I have lots of Latin and English and some French, so it's actually doable. Anyway, so I'm just curious what you think about this, like big language versus like small regional language, because it seems to me that on the one hand, you have this obvious utility if you learn a language like Spanish, but on the other hand, I don't know, it feels like that misses something about learning a language, just learning something because it's useful. And I would imagine if I were to learn something like Catalan, it would be much more, obviously not useful outside of the area, but like in the area, it would be way more useful and it would be super cool. Yeah, I'm curious just what you think about that.
So obviously I'm going to be very biased because I'm very proud of Catalonia. But I just made a book recommendation based on just sometimes doing things just for fun.
Yeah,
everything has to have a utility. So if you're going to read all of these books that are going to teach you something with the same ferocity that you're going to read the fun books, then great, even better. You get double benefit. But the reality is that most people don't. So if you're gonna try to learn Spanish, but you're gonna do it half-heartedly because you're like, that's not really, like, it's like, just from how you talk about it, you seem so excited about learning Catalan, and Spanish is, it would be useful. If that's how you really feel, then, learn Catalan.
For context, like I have no connection to Catalan at all, right? It's just an example, basically. And I think, it's basically also like I've lived in like Northern Europe most of my life. If I spend half a year in like going to a different culture, it might as well be by the beach in the south.
Catalonia is stunning.
Yeah. So, that's the thing. Like I have, again, like as I said, I don't know, I like watching football, so I guess I've seen Barcelona. But that's the extent to my connection to Catalonia. But yeah, I'm just curious because, I guess, okay, so your advice is just Catalan and Catalonia is awesome. Do it.
Well, yeah, obviously, in my very biased opinion, Catalonia is awesome. I would love to spend half a year there. I think it's an amazing place to be. Catalan is a really fun language. I think qua difficulty, it will be very similar to learning Italian or Spanish.
Yeah, so it's also like a language. Again, like I had Latin for like 6 years in school, English and a bit of French. So like, I think I could learn it very well in a year or half a year. If I again, that's like the thing I do in that time, right? Learning the language. Yeah. So like whereas Basque would be like probably not as fluent after any given time.
Yeah. And Catalan people are very appreciative of more people trying to learn.
Oh, that's true. They'll love that. Yeah. That's like a political statement almost. If I learn Catalan, I'm like making a statement. I'm not even aware of what the consequences are.
Obviously, if you're looking for utility, if you're looking for long-term impact on your life, Spanish will be much more useful. Though you never know. Barcelona is a great city.
Yeah. I actually found out that
you never know.
Did you know that they also speak Catalan in a non-Spanish or French-speaking country?
No, I didn't. Where?
A tiny area in Sardinia, so Italy.
Oh, yeah. I mean, I'm not surprised. Yeah, because
there's just like one small coastal area where they speak apparently some Catalan.
Yeah, I'm not surprised. I've tested this with my Italian friends, and if they speak Italian and I speak Catalan, we do understand each other.
Okay.
And that's not necessarily true with my Spanish friends. So
really?
Yeah, it is oddly. I don't know if this is actually based on if you analyze the language, you get the same result. But just based on anecdotal data, it seems to be closer to Italian than Spanish.
Oh, that's interesting. I guess. Yeah, I guess geographically I wouldn't have guessed that.
No, geographically it doesn't make any sense at all. So yeah, I don't know if this is true for real, or my Spanish friends were just not trying hard enough.
Yeah, they were like, I'm not gonna even try.
Yeah, but yeah, in my very biased opinion, of course I think you should learn Catalan.
Okay, yeah, we'll see. I mean, again, this is like a few years into the future. Basically, I guess like, yeah, I think something vaguely Italian or Spanish would make the most sense in terms of me actually learning it very quickly. And the language, I mean, it's weird with Spanish. I feel like I can understand a lot of it, especially considering I've never learned a word of Spanish. It's really shocking to me how, at least on first sight, easy it is. Literally 2 days ago, there was a guy here who needed directions, and his English was terrible. So I said, which language do you speak? He's like, Spanish. And I was like, yeah, I don't speak Spanish. And then basically he went to like Italian, no, Portuguese, no. And then I just spoke some French and he spoke some Spanish, and I managed to give him directions. Like it worked out pretty well.
It works out. No, I mean, within the Latin languages, there's a lot of overlap. Like you make it work. So, but that's really cool. We can make the next podcast in Catalan.
Okay, that's a deal. Yeah. Yeah, exactly. Yeah, that's okay. That's going to take a while. Okay. Anyway, thanks.